Hello, I'm Manfred Auer. I'm Director of Physical Analysis at the Joint Bioenergy Institute. I'm also a staff scientist at the Lawrence Berkeley National Laboratory. Most biological processes happen at the level that you can't see with the naked eye. So you need a microscope to look at those processes. When we look at the nanoscale, electron microscopy is your method of choice. For example, if you want to know how plant cell walls are organized, we need to use an electron microscope. Or if we want to know how microbes are efficiently degrade lignocellulose, then we need to use electron microscopes. We typically do this by freezing the sample instantaneously down to minus 180 degrees Celsius, and then we cut sections at very low temperature and keep the sections frozen, and then we perform what's called cryo-electron microscopy or cryo-electron tomography. And that visualizes the sample at that low temperature and keep the sample frozen and therefore in its native state. Typically, a sample preparation would be taking a few hours, but imaging can take up to several days, and the image analysis is really what it all comes down to. That takes us many months, typically up to a year or two. We also, at the end of this process, we build a model into our data sets, and that whole process can take up to a year or two. It requires years of training in electron microscopy and good background in computational science. We work with all divisions at JBEI, and we provide the high-end imaging support that they need in order to move forward with their respective projects. The question is, where are we going with this research? And one of the key topics is to push the envelope of what currently bioimaging can do. And one of the key concepts here is to integrate multiple imaging modalities into an entire workflow and to go across multiple scales. So we are not just able to look at macromolecules, we're not just able to look at organelles, but we go all the way to cells and even tissues. So this concept of integrated bioimaging is really one that's emerging now and requires a lot of method development, which we currently and actively doing. We can visualize for them cells that are producing biofuels. We can visualize plant cell walls. We can look at the altered plant cell walls, and you can see what are the properties and how they have changed. We have determined the first 3D architecture of plant cell walls. For decades now, people have thought about what the cell wall actually looks like, and there are several models out there that have been proposed based on some biochemical analysis. We are now for the first time have the actual structure of the plant cell wall. That's very exciting. We also looked at termite hindguts and see how the microbial communities in the termite hindgut degrades wood particles, lignocellulose. And we found that while there are about 200 species of bacteria live in the gut of the termite, there are only a few that actually do the lignocellulose degradation. So we think that is very powerful because we now can learn how a system that has evolved over the last few billion years actually does it most efficiently.